So within this video, we're going to be looking at some parts of 2.7, and the parts which we are looking at are DNA replication, and we're adding to that this extra knowledge here, 7.1, which is the higher level version of DNA replication, which is clearly much more detailed. If we look here, first of all, we've got 2.7 being split essentially into two parts. We've got the DNA replication part, and then over here, these two processes, transcription and translation, are all about the synthesis of a protein. And remember that within DNA, we are encoding the information that's required to make a protein, make a specific polypeptide. So this idea of the one gene, the one polypeptide is synthesized from, and that's the central dogma theory of biology. But within this video, we're just going to concentrate to start off with on this part here. And what we also need to appreciate, and the essential idea at the beginning here, is that genetic information in the DNA must be copied in an accurate way. So when we make new DNA, we need to make it so that it is exactly the same in terms of the base sequence as the existing DNA. If that goes wrong, what we end up with is a mutation, which we don't want to have. If we've got mutations, that can impact the reading of that information and the transcription and translation and the coding of that protein. So DNA replication must be done in a very, very accurate manner. When does DNA replication take place? Well, we should know this already, that DNA synthesis or DNA replication, same thing, happens in interphase in the cell cycle, that so-called resting phase, which is not really resting. It's got a very high energetic demand there, and DNA replication is going to happen just before we have nuclear division, either mitosis or meiosis. In this image here, what we can see is something called a polysome. This is not relevant until we look at this part up here, transcription and translation, but you could be given images, electron micrograph images, and be asked to identify them, so that's worth putting on your radar there. And if we look at the syllabus statements, it's these three that we're going to focus this particular presentation on. We come back to these later on. So to begin with, I'll just, there's more. These syllabus statements here as well we're going to look at. And what I'd like to do is later on this week, or possibly the beginning of next week, I want Brian, sorry Brian, this is you again, I want you to look at this idea here of Taq polymerase being used in a process called PCR, polymerase chain reaction. And polymerase chain reaction is when man artificially manipulates the process of DNA replication for profiling. So we'll come back to this later on, but Brian, again, can you make note of that? In a similar way, Jeremy and Eunice, what I'd like you to look at is the analysis of Messelston and Stahl. And Messelston and Stahl were individuals that put forward evidence to support this idea that DNA was replicated in a semi-conservative way. And we'll look at what we mean by semi-conservative later on. But again, put that on your radar. FYI, this comes up quite a lot in past papers, so do make sure that you're clear with this particular experiment. Quick recap then, we've got this idea that DNA, deoxyribonucleic acid, is made of two strands, and the two strands are anti-parallel. We've got this one running from the 5' to the 3' direction, and then on the opposite side, we've got the 3' to the 5' direction. Remember that this molecule also is extremely stable because of the hydrogen bonds which exist between complementary base pairs, and we've got 2 hydrogen bonds between adenine and thymine, and 3 between cytosine and guanine. And remember that the whole length of DNA is actually super long, and that means that we've got an incredible number of these hydrogen bonds. So one hydrogen bond is a weak electrostatic attraction, but because we've got so many, the molecule of DNA is very, very stable. So what do we mean by semi-conservative replication? So if we imagine the original DNA here, this is the double strand in blue, what we mean is that when we produce two strands, two new synthesized strands, half of this is the original blue, and then the other half is newly synthesized daughter DNA. So think about what semi means. Semi means half. So half of this new strand is parental, the original DNA, and the other half is daughter new DNA. I don't know why it's called daughter, it's very sexist really. Now, conservative, what does conservative mean? It means that the base sequence that runs down the backbone of these two here is exactly the same as the base pair sequence here, which is exactly the same as the base sequence here. So semi-conservative means that when you produce newly synthesized DNA, half of it is going to be the original parental DNA, and another half is going to be newly synthesized daughter DNA, and the base pairs that run along the DNA molecule are exactly the same. Now, Jeremy and Eunice will unpack the evidence which supports this, but these were the three proposed models before Messelson and Stahl demonstrated that this is indeed what actually takes place. So we've got conservative proposal where you've got obviously the new DNA here and the original there. Base sequence is the same, but what we've got is this newly synthesized strand is entirely different and separate from the parental one. There was also thought to be this dispersive model here, and as you can see that we've got little bits and bobs all the way through here, and this is meant to represent the first generation and then the second generation. But DNA synthesis is semi-conservative, and we know that this is the case because of the evidence that came from Messelson and Stahl. So now we need to think about how does it do this semi-conservative replication. And there are many enzymes involved in this process. The first one that you need to appreciate is helicase. And all enzymes end in A's. Remember that all enzymes have a very, very specific active site, and the substrate is going to fit into that active site to cause induced fit. When we have induced fit, that lowers the activation energy, which allows the reaction to proceed. Remember that all enzymes are in small quantities because they can be used over and over again. Remember that enzymes are sensitive to pH, they're sensitive to temperature. Remember that enzymes can be inhibited. So go back and just make sure that you're clear with all the work on enzymes and contextualize this. So this is just an image which represents a helicase, and as you can see, it's a complicated polypeptide, multiple polypeptides, so it's the quaternary level of protein structure that we can see here in this globular protein. So what does helicase do? Helicase unzips the DNA molecule. So if you imagine we've got the DNA molecule here, we've got hydrogen bonds between individual bases. Remember, guanine, adenine, cytosine, guanine. And what is going to happen is that helicase is going to move from what's called the origin of replication, and it's going to break those hydrogen bonds and unzip the parental DNA. Energy is required to do this, so you need to be aware that this is an energetic process. Now, as the DNA is unzipped, they then become referred to as parental strands, and these parental strands are going to act as a template for new nucleotides to be brought into place. And the enzyme that does this for 2.7 is this enzyme here, DNA polymerase. Now, we're going to look later on at the fact that actually there are multiple forms of DNA polymerase, and on the higher part of the course, we're going to look particularly at the actions of DNA polymerase 3 or DNA pol 3 and DNA pol 1. But for now, let's start at the beginning. Let's think of it quite straightforward. We've got DNA polymerase, and again, the DNA polymerase molecule is a quaternary level of protein structure. It's very specific in terms of its active site, etc. And here is an image that shows you what happens as DNA polymerase works to create the complementary strands. Now, if we unzip the DNA molecule at the parental strand, first of all, let's think that this one is going to be the 3' end. So this one, this newly synthesized strand, which is being synthesized in this direction, must be the 5' end. Now, DNA polymerase can only work in one direction. It can only work 5' to 3', and when we say it's working 5' to 3', we're talking about the 5' end of the newly synthesized strand. Take care, because there are many images online which are very misleading with regard to this. So we're going to use DNA helicase to unzip the original parental strands. Then we're going to helicase does that. Then we're going to have polymerase enzyme, which is going to bind, and it's going to add these nucleotides in place. So if this is adenine, it's going to be guanine, cytosine, adenine, guanine, cytosine, thymine. It's going to bring that nucleotide with the complementary base into position, and then it's going to bring the next one, and the next one, etc., etc. So DNA polymerase is bringing in the correct base to pair with the base, which is here. But remember, this is not just a base, this is a nucleotide that's brought into position. And on this particular strand, this is going to be continuously replicated. It's going from the 5' to the 3' direction. So if we think of this strand, we've got to make sure that we unzip it, and it's going to go again, because if that's the 3' parental end, this is going to be the 5' parental end. So this one is going to be copied in this opposite direction here. Now there's a problem, because obviously this is not yet unzipped. So this helicase molecule is still unzipping. So what we're going to find is that we have continuous replication on this side and discontinuous replication on this side. So it's going to do that little bit, then it's going to need to wait for DNA helicase to unzip it a bit more, and then it'll copy that little part there. And when we look at the higher level stuff, this becomes much, much more important. So we've got 3 nucleotides being brought to the DNA template strand by DNA polymerase, and we've got this idea of complementary base pairing. But can I again underscore that these are not bases, they are nucleotides. Now, when we look at the triple stuff, we're going to add more detail to this, because actually what this molecule is not just a nucleotide, it's got 2 extra phosphates added onto it. So we've got what is essentially a triphosphate nucleotide, and it's actually called a nucleoside triphosphate for reasons that we're going to come to. But for 2.7, we've got helicase unzipping the molecule, and then DNA polymerase bringing nucleotides into place using complementary base pairing, and it works in the 5' to 3' direction. It's going to work continuously on this side and then discontinuously on this side as the helicase unzips. Another detail to bring to your attention is the fact if we're bringing that nucleotide into place, we then need to join adjacent nucleotides. And remember, this is a phosphodiester bond. So not only is DNA polymerase bringing the nucleotide into place, the correct one, it's also forming this phosphodiester bond here. And there's some text to support that just here. And the last thing the DNA polymerase does is it proofreads that these complementary base pairs are correct. And if they are correct, that means that you're going to have these being equidistant apart from each other all the way along the strand. They're going to be parallel. And this slide again is reinforcing what we've already said, but we've got this idea of DNA replication moving in the 5' to 3' direction. And then we've got the phosphate bond is this one that's going to be produce the phosphodiester bond, pardon me. So we're bringing these into position, forming that phosphodiester bond. And then we've got the third idea. We've got these little discontinuous strands being produced on this one, and we've got continuous copying here. This is reminding you that we've always got a purine and a pyrimidine. So what we're going to think about here is that we've got DNA polymerase, which is copying continuously, no problem. Then on this side, we've got discontinuous copying. And what you're going to end up with is a little bit of a gap between these two. And that gap needs to be then sealed up. And that brings in the third enzymes, which is called DNA ligase. And finally, we've got this slide here. This is looking at, it's the same one really, but just reminding you that the 3 enzymes involved are helicase, DNA polymerase, and DNA ligase. Now, the function of ligase will become much clearer when we look at the triple level material in a little while. This next slide, if you jump into the Google Classroom, you'll see this PowerPoint, and you can click on these links, and I would recommend that you do have a look at these links. But Brian is going to come back and present this to us at a later stage. And this is, again, to do with Brian's presentation. Here we've got the analysis of Messelson and Stahl, and Jeremy and Eunice are going to come back and present this to us. And again, I would strongly recommend that you look at this link in the Google Classroom, and they will take us through some past paper questions which are associated with this work as well. Then we need to start thinking about the higher level stuff, and I'll put that in video 2.